How's everybody doing today? Welcome to class. Good to see you, Melanie. Good to see you, Andre. Hopefully we'll have a couple of other people join us, but if not, it'll just be us. Can you hear me? How's my sound coming through? Are you getting any audio? You should be. I hope so. All right, fantastic. Sleepy but good. I'm doing good. Another day, right? Okay, fantastic. I'm not using my microphone today. I wanted to see if I could get away with using the system sound. All right. Cool. Looks like we have a few more people here. Who's joining us? It's good to see you. 3 extra people. Who's joining us? Say hello if you like. One of those types of days over COVID. I know, right? All right. The gyms have finally opened back up, and I really want to go, but I'm afraid to, I was thinking maybe I'll go like at midnight because nobody's ever at Planet Fitness at midnight, and then I'll really wipe down one machine and just work out on one machine the whole time. Hi, Angeline. Good to see you. Hi, Tyler. Good to see you, too. Fantastic. I wonder if Erica Schultz is here today. We'll see. Okay. How's school going? Right, I hate to fight the people well, this is back before COVID, but I always hated fighting 1,000 people for a couple of Stairmasters, Oh, yeah, you don't want to come to Psych 150. You want to come to Developmental Psych. It's weird, I have so many students. I recognized your name and you were there today, and you felt oddly out of place, but I didn't know why. We were talking about consciousness today, and meditation and flow theory. All right. It looks like it's 1 o'clock, so let's go ahead and get started. Did you folks have a chance to watch the videos that I sent earlier today? How about that TED Talk with the visualization of prenatal development? And Melanie, are we talking about your strong suit this week? Aren't you, didn't you work in an OBGYN for a while? It was amazing and just fascinating how they can visualize that stuff. It really allows you to sort of imagine how well we can begin evaluating a child even before they're born. Sonography, I guess, and Mel, you might be able to speak to this better than me, how well you can visualize a baby using an ultrasound, right? You can really build pretty complete pictures of the baby, I think. You're working in a maternal fetal medicine office. Okay, well, here's the deal. I'm going to do the talking today. But Melanie and Angeline, you are professionals. So if you want to add anything, correct anything, or anything else, you should feel like being experts today. Okay? I probably don't know nearly as much as you do, so go ahead and feel free to help me out today. By the way, I hope you like my background. I changed it to the most elegant shade of blue that there is, Carolina blue. So today we're going to talk about the 3 periods of prenatal development and what occurs in each stage. And so for the exam, I'm going to ask you to be able to tell me what happens in the germinal, and the embryonic and the fetal periods. We'll talk a little bit about how the placenta is designed and what its purpose is. I'll talk a little bit about prenatal brain development, and then we'll talk about teratogens and teratology. That's what we're going to talk about today. And then on Thursday, we'll talk about the settings and methods for childbirth, Apgar's tests, and what it means to be preterm, small for date, low birth weight, as well as the consequences. And Melanie and Angeline, I expect both of you to give me lots of information, especially on Thursday. Yeah, so what can you give us a little bit of each of you if you have something to say about ultrasounds, I'd be interested in hearing what you can tell us about those. Hi, Lorena. Good to see you. Do you have any experience? Oh, yeah, I remember one time when we saw X, Y, or Z, or we use it to do this or that. I would be interested in hearing that. So pregnancy is 40 weeks after fertilization of the cell, or actually, 40 weeks from the previous menstrual cycle, correct? And so when we talk about prenatal development, it's typically broken down into 3 parts. So the first thing we're going to talk about is how we develop even before we're born. And so we have billions upon billions of cells that develop from one fertilized egg cell in the space of 9.5 months. All right, so what I want you to know is that the first stage is called the germinal period of prenatal development. It's the first 2 weeks from fertilization of the egg cell to implanting of the blastocyst in the uterine wall. So the blastocyst is the fertilized egg cell that has developed an inner part, a blastocyst and a trophoblast on the outside that is going to become the placenta. And so what you're going to see in this picture right here is the egg cell comes out of the ovary. It's fertilized typically part of the way down the fallopian tube. It develops, splits into 2 cells within 36 hours. Within 48 hours, it doubles into 4 cells. After 3 days, you've got 16 to 32 cells. And then, like I said, it begins to differentiate into a blastocyst and a trophoblast. The blastocyst is the baby. The trophoblast will become the placenta, and that impacts into the uterine wall. Any questions so far? Now, so you get this disc, this dark disc called a placenta, and then you have a cord, an umbilical cord, and I think it has 2 arteries and 1 vein, if I'm not correct, which connects the baby's blood system to the placenta. Right? Okay, 2 arteries and 1 vein and connects the baby to the placenta. The placenta is a disc-shaped group of tissues which implants into the uterine wall. Now, what you're going to find in the placenta is that the baby's blood vessels and the mother's blood vessels twist all around each other, but they do not actually connect. So the blood systems are 2 independent systems. And what this allows to happen is for smaller molecules to ooze from the mother's blood system into the baby's blood system, and smaller molecules to ooze from the baby's blood system through the capillary walls to the mother's blood system. But it acts as a filter, keeping larger molecules like red blood cells, bacteria, and other molecules out of the baby's system. So think of the placenta as a filter. All right, and what happens? Small molecules like salt, water, oxygen can diffuse between the capillary cells from the mom to the baby, and molecules like carbon dioxide and intracellular waste can diffuse from the mother to the baby. But it acts like a filter, if you will. Now, what happens with psychoactive drugs, and we might talk about them a little later today, is psychoactive drugs typically, or any drug that is a teratogen, is small enough to fit through these interstitial spaces and seeps from the mother's blood into the baby's blood. So when we try to figure out which molecules are dangerous to the baby and which molecules are not, it typically depends upon the size of the molecule, because this system is designed to scrape out large molecules and keep them from messing up the baby's developing system. Does that make sense? Have I described that poorly enough? All right. Okay, and then So you've got an amniotic sac, which is that clear fluid in which the developing baby floats. And when they do the amniocentesis, they stick the needle into this amniotic sac and take out some of the fluid. Okay, hope it makes sense. All right, so around 2 weeks, around 14 days, what you're going to start to see is a neural groove begins to develop. So this little developing embryo is going to develop into 3 sort of layers of cell. It's going to have cells on the inside, the endoderm, and that's going to create your digestive and your respiratory system. Then you're going to have a set of cells that develop around the outside called the mesoderm, and that's where your circulatory system, your bones, and your muscles are going to develop, if you will, outside of your guts and lungs, right? And then on the very outside, you have the ectoderm, which is going to develop into your skin and your spinal column and your brain, all right? And so what you're going to see in this set of cells, after it implants in the uterine wall, what you're going to see in the group of cells is you're going to start to see a little fold in the back called a neural groove. And it's going to start about 14 weeks of age, and it's going to get deeper and deeper until the top folds over the top, and you're going to get this thing called a neural tube that develops along the outside, the back of the little developing embryo. And this neural tube is going to become the spinal column and the brain of the baby. Okay, and this development of this neural tube in the nervous system beginning to develop signals the onset of the embryonic period. And during this embryonic period, all of the organs in your body from 2 to 8 weeks begin to develop. Now, some of them are going to be done quite quickly, maybe 11 weeks of gestation. Some things like your brain and nervous system aren't going to be fully done until 25 years of age. But during this period, from 2 to 8 weeks, it's going to go from a little pocket of cells that looks like a ball to the beginnings of a human being that you can begin to identify. So the circulatory system is going to start, the nervous system is going to start, the intestine, the digestive system is going to begin, and all of these organs are going to begin to form. And we typically call this process organogenesis. The little arm buds and the little leg buds are going to form. And when we talk about teratogens and their effect, the effect of teratogens a lot of times depends upon the timing. So if you have some sort of drug exposure during the 2 to 8 week period of gestation, what you're going to get is structural malfunctions in your organs. They're not going to grow right. They're going to be missing parts. You may not grow your full arm or your full leg. But if you get a teratogen, an exposure to a chemical later on, what may happen is the structure of the organ may be all right, but the function of the organ will be damaged. I hope that makes sense. Now I want to talk a little bit about neural development because your book does. If you'll notice right here, we've got a neural groove developing in the ectoderm of the embryo right here. Right now, the neural groove then turns into a neural tube. Starting 15 to 18 days, and this neural tube is going to become your spinal column and your brain. Now, your book talks about the fact that sometimes the neural tube does not close up and this little system doesn't form. Which can result in anencephaly, which is the baby sort of born without a brain, and spina bifida, the holes in the spinal column. Research has shown, and doctors feel fairly confident, that folic acid or vitamin B is a good protectant factor against neural tube defects. So what they typically tell expectant mothers to do is to begin taking folic acid. Now, it turns out most females don't find out they're pregnant until how far along are they? Ladies, I would hate to speak for you. How far along do you get before you know before you take that pregnancy test? Ladies, if somebody wants to type in 5 weeks. Okay, so 5 weeks. So you're already way into your neural tube development. So the bad thing is a lot of times females may get pregnant not expecting to, and you're 5-6 weeks into neural development or 3 or 4 weeks into neural development before you even know about this. So it can be sort of a tricky thing. I think if you're of childbearing age as a female and you're in you're in a potential partnership where you might want to have a baby, it's probably a good idea just to take folic acid as a general rule. Oh, it's no problem. Glad to see you showing up, Eric. Good to see you, Erica. Good to see you. Now, here's the crazy thing about neurons. You have about 100 billion. I don't know who counted, but let's say you have billions and billions of neurons. All of these neurons are really created in 2 parts of your brain. This area beneath your hippocampus, in these areas beneath your ventricles in your brain. All the brain cells you will ever have are created in 2 places. And this process of creating neurons is called neurogenesis. Now, here's the part that just blows my mind. All of these neurons then migrate from these 2 places to the correct location in the brain. And I read this article by this fellow named Pasco Rakic, I think, and he shows that this migration occurs along chemical trails that these neurons follow. So these neurons actually migrate to the correct place in their brain. And then later on in the embryonic period, they begin branching out and sending axons in a programmed fashion to another part of the brain. So if you remember in Psychology 150, I suggested to you that neurons are information communicators. They're stimulated in one part of your brain and they send an axon out to another part of your brain. And if this part of your brain is stimulated, it causes another part of your brain to be stimulated or inhibited, right? All of this connecting, neural connectivity, is done before you're born, and it's all to a certain extent genetically programmed. It's kind of amazing. So neurons are born, neurons migrate, and once they get there, they send out their root, their axon, to another part of the brain. And this is all done in an epigenetic fashion. Now, what happens after you're born, when your parents play with you, when you stimulate, the part of the neuron that grows when you're stimulated by your mom or playing toys is the dendrites. So the cell bodies are already in place. They already sent the axon out. But then the only part that experience really changes is the growth of the dendrites and terminal buttons towards one another. And that occurs, and we'll talk about that next week when we talk about physiological change in infancy. Am I making sense to anybody? Are you following me? Have I bored you yet? Yes, sir, Eric? Do you have a question? 6 to 8. Okay, cool. Now, so that's sort of nervous development. Okay, good deal. I forget where the dendrites are. What do you mean you forget where they are? That's an interesting question. You know what? If you don't mind, can I go to the whiteboard really quickly? I don't know if you can hear me because I don't have my mic on, so let me know if you can hear me. Can you hear me well when I get over here? Can you hear me? Okay, so this is the neuron. This is the cell body. This is the axon. Remember, axons are covered by myelin tissue, so they look like bumps like this. And they go all the way down here, and they have these things called terminal buttons. terminal buttons down here. And these little terminal buttons have chemicals. And so up here, attached to the cell body, are all these hair-like structures called dendrites. Okay? And so what happens is the neuron moves to where it's supposed to be, and then this axon grows to a different part of the brain. And then when you stimulate a neuron, these dendrites start to grow. And so as you play with this child, these dendrites will start to get more complex. This occurs after birth. Okay? Does that make sense? This and this occur before birth, and this, the growth of the dendrites, occurs after birth. Is that the question you were asking, Eric? Blooming and pruning. Yes, we're going to talk about blooming and pruning. Melanie remembers her developmental chapter from last semester. We're going to talk exactly about blooming and pruning. Now, the weird thing is, Eric, do you know have you ever heard the terms gray matter and white matter? Does everybody know the term gray matter and white matter when we talk about brain areas? Okay. Most definitely. Okay, which is on the outside of your cortex, gray matter or white matter? Dendrites are the gray matter. Absolutely. Dendrites and cell bodies are the gray matter. And the axons are in the white matter, because the white matter is nothing but wires. Think of it as wires connecting different parts of your brain. Hey, what's up, D? Good to see you. Okay. So, just want you to be familiar with the idea of prenatal development. Neural groove, neural tube, closes up, vitamin B to make sure that happens so that you don't have spina bifida or another neural tube defect. Neurons are born close in two places in the brain. That's called neurogenesis. They prenatally go to a genetically determined spot in the brain, and then they send out a root, and that's all genetically programmed. All right, the fetal period is a 7-month period between 2 months after conception and birth. So all of the structures are created. So you see here on 8 weeks, you see the little eye, you see the head, you can see the arms and the legs of this funny-shaped little creature. It is just about finished the embryonic period, and it is moving into the fetal period. So you can think about the embryonic period from 2 to 8 weeks is the organogenesis part. Now, after 8 weeks of gestation, the baby is now called a fetus. And this is the stage during which the baby is going to gain the most weight. All right. If you looked at the baby video of them doing the cleanup of the newborn baby, you notice the vernix caseosa all over the baby. Did you folks see that nasty, white-looking, flaky stuff that was on the baby? Did anybody remember seeing that? Maybe you remember that. Those of you saw that. I don't know. What do you use to clean that up, Melanie? It looks like that stuff is hard to get off. And you'll actually also see that hair grows on the baby actually during the fetal period. This very fine hair is called lanugo. Now, from what I understand, babies are viable at about 6 months of gestation. Melanie and Angeline, in your experience, does that number sound about right? And I'll wait for your answer. Hopefully everybody hears me. Okay. So, yes, okay, 6 months. So typically underneath 6 months. Okay, all right. So we have some nursing students here. You know, sometimes you'll read something in the textbook or look at it in articles online, but then the reality will be something a little bit different. So I always like to ask the practitioners, because you're the ones that know. Now, if you'll look in your book, you'll notice that this is a summary of what we've been talking about today. We talked about the 3 periods in green. You've got the germinal period, let's see, wait a minute, hold on. We've got the, well, actually, I take that back. It shows you these different stages. This is sort of what I want you to know for the homework and the test. I want you to know what the 3 periods of are, when the time frames are, and what major occurrences happen in them. And I also want you to know a little bit about how neurons develop and move to their correct place. Any questions so far? Okay. Now let's talk a little bit about teratology, which is the study of hazards to healthy prenatal development. Remember, we talked about... Ooh, am I freezing for anybody else? I'm wondering if that's just you. Is it still freezing for everybody? The coating protects your baby's skin from getting pickled by the amniotic fluid. Well, that sounds gross. Is it easy to clean off? It looks awful, Melanie. Okay, I'm sorry about that. I wonder why my CPU usage is up kind of high. Let me go ahead and cut some things out. Maybe this will help. Okay, I don't know if that helped at all. All right, so remember we talked about in week 1, the whole point of developmental psychology is that it increases the length and quality of the lifespan. Yes, it does, Melanie. And so learning about healthy development allows us to improve the quality of human life. And remember, I told you the whole point of this class, what you're going to get from this class, is how to raise healthy, happy children and to live your best, longest life. And so that starts with prenatal development. You folks have all heard doctors talk in newspaper articles and PSAs talk about the fact that there are some things expectant mothers shouldn't do, some drugs they shouldn't take, some substances they shouldn't consume. And the study of this is called teratology, the study of hazards to healthy prenatal development. A teratogen, a little monster, is any agent that can cause a birth defect or negatively alter cognitive and behavioral development outcomes. Typically, we think of teratogens as being external molecules, and in this case, these molecules are small enough to penetrate the filter of the placenta, okay? But experiences, stress, other events that happen to the mom, there are genetic and biological qualities that can be teratogenic. But any harmful agent that can negatively affect a developing baby is collectively called teratogens. All right? And so teratogens can do lots of things. They can stunt growth. They can cause organs not to grow. They can cause organs not to function properly. They can affect brain development. They can affect growth. Absolutely. And you know what, it's kind of, I almost feel ridiculous going over this part of the chapter because, yeah, I'm going to tell you folks, hey, don't drink alcohol. And we all know that, but I'm still going to have to hit it. You know, a funny thing, folks, when my mom was pregnant with me in 1969, she was a heavy smoker. And when she would go to her OBGYN appointment, after the appointment, her and the doctor would sit there and share a cigarette. My mom would actually smoke a cigarette with the doctor. Now, this was the Deep South in 1969, but that's how they did it back then. And you know what? Nothing's wrong with me at all. You know, so. Yeah, clearly different times, man. So what determines whether or not a teratogen is going to affect you? It depends on several different factors. It's the dose. It's the timing of the exposure. And you know what? To a certain extent, it may be your genetic susceptibility. So, there are lots of things that determine whether or not a teratogenic substance is going to affect a developing mom. So here we go. This is a picture sort of prenatal development, what we've been talking about. You'll notice that the pink on the left side, if you look at this bar running from left to right, you've got this big slope here. This slope represents the 3 stages of prenatal development. You've got a blue germinal period on the left. The organogenesis is the purple embryonic period. And then the blue stage on the right is the fetal period. And so what you're seeing right here is sort of a graphical representation of pregnancy. And what you see here is the zygote implanting into the uterine wall. And then what you see here during the embryonic period is sort of a graphical representation of every system that begins developing during pregnancy. So the heart and the central nervous system are going to begin developing in week 3. And you'll see that the central nervous system has a bar. And you'll notice that this bar starts at week 3 and continues all the way to the end of this chart. And it's red for a little while, and then it's blue for a little while. The red is when that organ is developing, and it is most sensitive to teratogenic exposure. So you'll see that the central nervous system begins developing in week 3. The heart begins developing at the end of week 3. The ears begin developing in week 4. The eyes, arms, and legs begin developing at the end of week 4, the beginning of week 5. And so what you'll see is that depending upon when you take the teratogen, it's going to have different effects. Typically, from what I understand, is teratogenic exposure during this organogenesis period, during the red period, actually damages the structurally damages the organ. Whereas teratogenic exposure during the blue period may result in functional problems, structural problems versus functional problems. Does that make sense? I hope that makes sense. Okay. So common teratogens, alcohol, fetal alcohol spectrum disorders. If the mom drinks too heavily, children can be born with deformed facial features, heart problems, and learning and behavioral problems. Your book even talks about a research study, it's almost unbelievable, in England, where they found that the average lifespan of children born with fetal alcohol syndrome at 43 years of age. 43 years of age, that's 30 years less than somebody not born with fetal alcohol syndrome. The book suggests that fetal alcohol syndrome is associated with early deaths from suicide, accidents, and drug overdoses. Then Melanie, you mentioned nicotine, and that has been shown to be associated with low birth weight, attention deficit disorder, hyperactivity disorder, impaired brain development, and attentional learning difficulties. So, I've often heard that probably one of the biggest risk factors to children is low birth weight. Melanie, Andre, or Angeline, do any of you want to talk about your experiences with low birth weight babies at your clinics? Can anybody add anything? What is the big scary part associated with low birth weight? And I'll move on. It turns out that teratogens are not only illegal or recreational drugs. Prescription drugs can also be teratogenic. Your book suggests that some antibiotics, some antidepressants, hormones, and acne medication may also be teratogenic. There was a prescription drug that was given out in London, very famous case from the early 60s. It was a drug called thalidomide, which was given to expectant mothers as an anti-anxiety, and it had horrible teratogenic effects on developing babies. In fact, I think they only used it on the market for less than 6 months. But prescription drugs are not safe just because your doctor gives them to you. And usually what they're going to ask you whenever you get a prescription is they're going to ask you if you are pregnant or plan to get pregnant. Your book talks about caffeine, meth, cocaine, marijuana, and heroin as all being teratogenic as well. The weird thing, we're going to watch a video for tomorrow's class about mothers giving birth to babies addicted to heroin. And from what it sounds like, one of the main problems with heroin use, I'm not sure if it necessarily destroys babies' functioning as much as it causes them to be born addicted to heroin and to have central nervous problems associated with coming off of heroin. And in fact, in the article, in the video that we're going to watch, they actually suggest that mothers not kick heroin during pregnancy because the stress of having this drug removed from the system can lead to premature birth. And they say that the most dangerous thing for these babies is being born premature, even more than being born addicted. And we're going to talk about a method for treating babies that are born and have to go through heroin withdrawal. Any other questions? Now, there's, has anybody, have any of you ever had to take Rogam? Any of you moms ever had to take Rogam? So it turns out there's the mom's blood type can actually be teratogenic. So if the mom, that is a concern because it increases complications, baby have a harder time eating, gaining weight, have a harder time fighting infection. They can also have a hard, and she's going to complete that sentence, I'm sure, regulating their own body temperature. Okay, so one of the main problems with the heroin exposure and removing the child from and having the mom stop is that you could cause the mother to have the baby early. The low birth weight leads to all of the problems that Melanie just mentioned. Thank you. Now your book also talks about blood incompatibility. So you have an Rh factor on your blood. So not only do we have blood types A, B, and O, but we can also have a positive and negative, A positive, negative, positive, O negative, O positive. That's called it's called an Rh factor, and it's antibody that you have on your blood cells. From what I understand, if you are Rh positive, it means you have the antibody, or the protein, is it? Help me out. Wait, hold on. Creates the Rh factor, excuse me. It's just an Rh marker. And what happens is if the mother does not have the Rh factor, if she's negative, A negative, and she has a baby that is A positive, that has the Rh factor, her blood will create an antibody to fight that Rh factor. From what I understand, that does not hurt the first baby. So if mom is an Rh negative and she has an Rh positive baby, that baby will be born fine. However, if that mother has another baby that is Rh positive, her system, because she's built up the antibodies, her system may attack the developing baby. And I think one of you mentioned that your baby was born with jaundice. I watched a little video on the Rh factor and this Rh incompatibility, and they suggested that it's not always noticed, and they talked about the fact that this can lead to jaundice. Rh has to get injections so the body doesn't attack the fetus, treating it like a foreign body. Yeah, and I think the drug that they give you, or the injection that they give you, is called Rogam. I don't know if that's the trade name, Melanie. You may know it by a different term, but your book's going to call it Rogam. And then there are all of these environmental and disease factors that moms can have that can affect the developing baby. And I'm probably not going to ask you about any of these specifically, so I'm just going to hand wave a little bit at them, if you will. All right. So that's all I had to say today. I wanted to talk about the 3 periods of prenatal development: germinal, embryonic, fetal period. What happens in each of those 3 stages? What's organogenesis? What's the pattern of neural development? What's vernix caseosa? What is the function of a placenta? And so then I want you to talk to me a little bit about the prenatal brain development. How does the nervous system development? Neurogenesis, neuromigration, and then sending out the root-like axonal structures. And then I want you to talk a little bit about what's important about teratogens, the timing, the dose, and the genetic susceptibility. And I'd like you at least to talk intelligently about the Rh incompatibility, heroin, and fetal alcohol syndrome as teratogenic situations. Does anybody have anything they want to add to this class? That's really all I have to say for the moment. Hopefully you'll come to tomorrow's webinar at 1 o'clock. We're going to be talking about teratogens and asking the question, what kind of culpability do you think parents who are born, who are addicted to drugs and take drugs while they have babies, what kind of culpability do they carry? And what would you do? I'd be interested in hearing your opinion about that tomorrow. All right, folks, you have a great day. I will talk to you on Wednesday. Take care.